So the first step of glycolysis is performed by hexokinase. So hexokinase performs the reaction, converting one glucose molecule and ATP into G6P glucose 6 phosphate, where the glucose has been phosphorylated on the 6th carbon, bringing in some ideas from our carbohydrates lecture, and we can count the number of carbons, right? On the screen, I've shown you kind of the three-dimensional conformational change that happens when this enzyme binds its substrates. So first, the enzyme has this like open-mouthed Pac-Man shape, and when the substrates, glucose and ATP, bind, this enzyme closes around them. This is important for two reasons. First, we are excluding water from the active site, so we're not just going to have random hydrolysis happening because water's there. But remember from our enzyme mechanisms class that we also need to be thinking about the proximity effects. Proximity. If the enzyme shuts down tight over these substrates, they can't go anywhere, so they're more likely to react with each other. And I want us to take a look at the mechanism happening here, because this is the mechanism that is similar to many mechanisms in glycolysis, where we have some type of phosphate transfer happening. It is simplified for us in the top left of the screen, but I'm going to draw it out in a bit more detail. Here's our ATP. I'm going to simplify the first two phosphates because we're only using the last phosphate in the mechanism. Okay, folks. So in this mechanism, what's going to happen is that a base from the enzyme is going to first abstract the proton from this OH group here. Let's recall, OH is going to be a weaker nucleophile than O-. Then that nucleophile, O- is going to go ahead and attack the biochemist's favorite electrophile, that thing that looks like a carbonyl carbon, which is the phosphate in this case. Phosphate only, phosphorus only forms 5 bonds at a time, so we need to break off this last bond. And what do we get? Our products a DP and this glucose molecule that now has been phosphorylated at carbon 6. And this mechanism is going to be common basically for any enzyme here that's performing a phosphate transfer. This is the first rate determining step of glycolysis. You will note that the reaction scheme I have at the top of the page does have equilibrium arrows, but note that the top arrow is much bigger than the bottom arrow, so we should more or less just assume that this is a unidirectional arrow, and you will see on the handout provided to you that this step actually has a single-headed arrow. What is the point of this first step? Well, folks, Glucose as a molecule does not bear any charge, right? It's a polar molecule, but it is neutrally charged. Popping this phosphate onto the glucose gives it a negative charge immediately, right? And charged molecules have a much more challenging time leaving the cell. They can't cross the membrane, for example, because the lipid bilayer is nonpolar in the middle, and something supercharged isn't going to cross through that nonpolar region. So in effect, phosphorylating the glucose traps it inside of the cell so that the cell can keep it and use it for energy. On to step 2, phosphoglucose isomerase. This enzyme takes the G6P that we just made and isomerizes it, I'm sorry you can't see this, to instead a 5-membered ring. So we're going from a 6-membered ring to a 5-membered ring, glucose to fructose. Now we don't know yet why this reaction is necessary, but we'll see very soon why we needed to do this isomerization reaction. Don't worry about knowing the mechanism. I have it on the slide here for you, for those of you who'd like to look at mechanisms. The third step in glycolysis is performed by phosphofructokinase. Oftentimes this is abbreviated as PFK. This enzyme takes fructose 6-phosphate and ATP and uses this ATP to add one more phosphate onto fructose 6-phosphate, giving us fructose 1,6-bisphosphate. So now there's a phosphate attached to carbon 1 and carbon 6. The mechanism here, folks, very similar to hexokinase. I bet you could draw it. Instead of having the hydroxyl group from carbon number 6 doing the attack, it's the hydroxyl group from carbon number 1. This enzyme catalyzes the second rate determining reaction of glycolysis. We will learn more about this step when we come together next time because it is the main site of regulation. Now, what is the point of this step? Folks, eventually what we need to do to glucose is create some high energy bonds on glucose that we can harvest to make ATP. Recall, the end product of glycolysis, we have to make to net ATP. In order to make those ATP, first we'll need to make some high energy bonds to hydrolyze to form that ATP. So this is a precursor. If we just pop some phosphates onto the molecule, let's make them high energy in the future. Next up, step 4 is performed by the enzyme aldolase. We are taking fructose bisphosphate, so this is the molecule with the 2 phosphates, but now you're seeing it in the linear form. And we are chopping this molecule in half into 2 3-carbon unit molecules, into GAP and DAP. This enzyme is important because in order to kind of maximize the energy that we can get from glucose, we need to split it up into 2 3-carbon units so that we can harvest energy, more energy from that molecule. Basically, we'll have now 4 ends to work with, whereas there's only 2 ends in fructose bisphosphate. That gives us more opportunities to create high energy bonds. But let's do a quick throwback. I told you we couldn't really discuss yet why this step was important. Step 2 is required in order for us to do step 4. In order to perform the reaction step seen here, we need the ketone. We need this carbonyl to be located at carbon 2 and the hydroxyl at C4. Remember that we switched from glucose to fructose. Glucose is not a ketose. Glucose is an aldose. which would mean that the carbonyl carbon would be at carbon 1. This reaction down here can't happen if we don't have the ability to do resonance at carbon 2. So step 2 is required in order for step 4, the cleavage of a 6 carbon unit molecule into 2 3 carbon unit molecules, because we need to have that carbonyl at 2, not at 1. The reaction doesn't work if that carbonyl carbon's not there, because we can't do resonance to stabilize the products. We don't see it here on the screen, but a cool note is that covalent catalysis is used to stabilize the reactive intermediate. You might understand why this reactive intermediate needs to be stabilized, folks, if you see that it bears a charge. And we know that many charged intermediates are stabilized. Step 5, almost halfway through, folks, is Triose phosphate isomerase. Okay, only GAP is used for the rest of glycolysis, but we just made 1 molecule of DAP and 1 molecule of GAP. So this enzyme takes DAP and changes it to GAP so that we can proceed with the rest of glycolysis. This enzyme does acid-base catalysis, and a lysine, positively charged, stabilizes negatively charged intermediates. This is a very common theme. And this gets us halfway through glycolysis. We have seen glucose converted into 2 molecules of GAP. So far, we've only seen an input of energy. We used 2 ATP along the way to phosphorylate this molecule and to chop it up in 2. Recall from here on out, each reaction that we see will happen twice because we have 2 molecules of GAP.